Excellent choice! Let's get started learning a bit more about lipids. So, what is a lipid? How would we define it? A lot of you may have heard lipids as synonymous with fats, but they're actually more than just fats. Generally speaking, lipids are mostly hydrophobic molecules, meaning they are mostly not soluble in water, and they are made up of a lot of carbons and hydrogens bound together. This contributes to their hydrophobic nature, and it causes them to aggregate or cluster together when they are placed in water. This forms what we call globules. This aggregating property, how these molecules all clump together when they are exposed to water, is really the defining feature of all lipids. Now, I am cautious to say mostly hydrophobic here, because as we will go over shortly, not all lipids are 100% hydrophobic. But they do still all spontaneously aggregate or clump together when they are placed in water. That is key. Now, thinking back to our chemistry of biology discussion, and knowing that lipids are mostly made of a lot of carbons bound to hydrogens, how could you have determined that lipids are all or mostly hydrophobic without being directly told? Moving on to the function of lipids, lipids have three main functions, which are long-term energy storage, membrane formation, and chemical signaling. Each of these functions is largely associated with a specific kind of lipid. Long-term storage is mostly associated with a type of lipid called fats, the one that most of you have probably heard of in some form or another. Membrane formation, though, is largely associated with a kind of lipid called phospholipids. And chemical signaling is largely associated with steroids, another type of lipid. So let's break down the structure and more specific function of each of these three main types of lipids, starting with fats. We're going to start by looking at the structure of fats, and then we will use this knowledge to dive into their specific functions. Sound good? So starting with the structure, fats have two main components: a glycerol, or 3-carbon polyalcohol backbone, as it were, and fatty acid, which are long hydrocarbon chains with carboxyl groups, tails. Now, remembering back to our discussion on monomers and polymers, we join our glycerol head, or our glycerol backbone, to these 3 fatty acid tails via a dehydration reaction. So we end up getting something like this. What you see here is what we call a triglyceride. Tri for the 3 fatty acid tails, and glyceride referring to the glycerol backbone. Now, there are 2 types of fats. We have our saturated fats and our unsaturated fats. Saturated fats refer to fats where within the fatty acid tails, the carbon atoms have as many hydrogens as they can possibly hold. They are saturated. So there are no double bonds found between the carbons, just single bonds, each carbon bound to a hydrogen. These fats are pretty linear as a molecule, and these types of fats are produced by animals mostly. Well, except some fish oils, those are not. But most animals produce this kind of fat, and they are known to be solid at room temperature. In contrast, unsaturated fats have 2 or more carbons in their chain that are double bonded together. Their fatty acid tails are not saturated with hydrogens. And these double bonds can cause the tails to have kinks or bends in them. These kinks keep the molecules from becoming really dense, which is why unsaturated fats are usually liquid at room temperature. These types of fats are also usually produced by fish and plants, with the notable exception of tropical plants like palm and coconut, but otherwise this is what you would get. Now, transitioning from structure to function, fats can be used for insulation or cushioning, but one of their main functions, in addition to this one that we mostly usually know about, is that fats actually serve as long-term energy storage. The many carbon to hydrogen bonds of fats, far more than that seen in carbohydrates, form a long-term energy storage for the organism that it's within. So for comparison, fats are about 9 kilocalories of chemical energy per 1 gram of fat, while carbohydrates equate to about 4 kilocalories of chemical energy per 1 gram of carbohydrate. So if you'd like to know a bit more about the chemistry behind this, behind why fats are so much more energy dense than carbohydrates, basically we can count each carbon to hydrogen bond as energy stored, and count each of these bonds as a net zero energy, okay? Now carbohydrates have roughly 1 carbon to hydroxide bond for every carbon to hydrogen bond. So roughly half of the potential energy content provides 0 energy. Fat consists of mostly carbon to hydrogen bonds though, so there is a lot of energy in fats. So when an organism consumes too much carbohydrates, in plants it's converted to starch, and in animals it's converted into glycogen, right? But that's for initial shorter term energy storage of carbohydrates in animals. For longer term storage, for example, if the animal keeps eating more and more carbs, the animal's body will then convert the carbs, those excess carbs, into fats for future use. Many humans actually gain weight as they grow older because the amount of energy that they need to survive decreases as age increases. However, they usually still eat the same amount of food because it's what we are used to. This means more carbs are being converted to fat, leading to weight gain. So diving into our next check-in, this is check-in number 27, so make sure you're keeping an eye out for 27. Which biological molecule can store more energy per gram of that molecule? Carbohydrates or fats? So that's fats. Now let's move on to discussing phospholipids. Phospholipids are similar in structure to fats in that they both have fatty acid tails as well as a glycerol backbone. But the one key distinction between these two is that phospholipids only have 2 fatty acid tails. Instead of having a third fatty acid tail, they have a phosphate group as a quote-unquote head to this structure. Now, why is this important? Well, because this structure actually relates directly to their function. Phospholipids are responsible for membrane formation, or forming membranes because of their unique structure. As we already know, the fatty acid tails are very hydrophobic in nature. And the phosphate group, or the phosphate head, is actually quite hydrophilic in nature. We call molecules that are both hydrophilic and hydrophobic amphipathic. Now, because phospholipids are amphipathic, that means that if a bunch of these are placed in water, we would expect all of the hydrophobic tails to aggregate together to avoid water, while the phosphate groups would all be facing outward since phosphate groups are hydrophilic and can interact with water. This is essentially how the lipid bilayer that forms the plasma membrane in cells came to be. In this configuration, all of the hydrophobic tails are protected from the water, while the hydrophilic heads are able to associate with water. Notice anything interesting about this image? Right, this is how the plasma membrane of a cell usually looks, right? And then inside we would have all of our organelles, but the plasma membrane, which is made up of phospholipids, protects the cell from external environmental factors. It keeps it separate from other things. And this is all because of the structure of a phospholipid. So diving into check-in 28. Go ahead and evaluate the structure on the screen and label the following molecule. Be sure to label what the name of the structures are, as well as whether or not they're hydrophilic, hydrophobic, or amphipathic. So that's phospholipids. Now let's move on to discussing steroids. Steroids are pretty unique in structure, but they are still lipids because they are hydrophobic molecules. They are made up of a lot of carbons and hydrogens bound together, and they spontaneously clump or aggregate together if you throw them in water. Now, before diving into it, I just want to make sure everyone fully understands my drawing on the screen here. This is basically shorthand for drawing out molecules. So everywhere you see 2 or more lines meet, like here, here, or here, these points represent where carbon is. Now, steroids are made up of 4 carbon rings with a hydrocarbon chain. This example that I have actually drawn up on the screen is reminiscent of our most common steroid called cholesterol. I think technically I'm missing a couple double bonds for it to officially be cholesterol, but it's very reminiscent of it. Now, cholesterol is present in the cell membranes and changes the fluidity of cell membranes. It is also a precursor to hormones, such as testosterone, estrogen, cortisol, and vitamin D. Fun facts, vitamin D is actually a hormone, not a vitamin. While most hormones are proteins, some, such as estrogen and testosterone, are lipids, specifically steroids. So in short, the function of steroids are that they play an important role in membranes, and they are involved in chemical signaling. So that's steroids. Let's dive into your final check-ins. Check-in number 29, list the functions of lipids. And check-in number 30, what do all lipids have in common? And that concludes our discussion on lipids. Awesome work!